The cells most important to the humoral response are the B cells, named so because they develop in the bone marrow. These B cells each have one of millions of distinctive surface antigen-specific protein receptors that are inherent to the organism's DNA. This is where structure relates to function. Since B cells mainly function to produce antibodies specific to certain unknown pathogens, B cells have different antigen receptors in order to recognize different potential pathogens. After the antigen receptor binds to an antigen, the B cell then differentiates into a plasma cell, which produces antibodies, a soluble form of the antigen receptor. In fact, naive B cells express antibodies on their cell surface, called membrane-bound antibodies. In this drawing of an antibody, you can see the basic components of it. It consists of a heavy and light chain, which are connected with disulfide bridges from the oxidation of sulfhydryl groups. All antibodies are alike in that their light and heavy chains all have a constant region, where amino acid sequences are all very similar among the receptors. This region determines the class of the antibody and does not vary within a given class. In other words, this area does not directly interact with the antigen. However, antibodies also have a region called the variable region, which vary extensively from one B cell to another. These variable regions in the light and heavy chains form the binding site for the antigen. The way that B cells originating from the same bone marrow with the same DNA can have different receptors is that in B cell development, there is a lot of shuffling of the portion of the DNA responsible for the variable parts of the protein receptors, the parts of the receptor, and eventual antibody that actually respond to the pathogen. After formation and maturation in the bone marrow, the naive B cells then move into the lymphatic system to circulate throughout the body. In the lymphatic system, naive B cells encounter an antigen and recognize it using an antigen receptor, a protein that is able to bind to specific molecules on a particular pathogen. This starts the maturation process for the naive B cell. Finally, the B cell engulfs the antigen. The portion of the pathogen that its receptors recognize is called the epitope. The B cell then digests the pathogen and presents a piece of the antigen on an MHC2 molecule. Now as we move someplace else in the lymph node, the activated helper T cell in turn activates a B cell with receptors for the same antigen. The B cell displays the antigen fragment on the cell surface with the class II MHC molecule again. Activated helper T cell with receptors for the displayed fragment binds to the B cell and activates the B cell. In most cases, both an antigen and a co-stimulator, which is usually a helper T cell, are required to activate a B cell and initiate B cell proliferation. This direct cell-to-cell contact is very important for B cell activation, and one antigen is likely to activate a variety of B cells since most antigens contain multiple epitopes. The activated B cell proliferates and differentiates into memory B cells and effector cells, called antibody-secreting plasma cells. Memory B cells provide future immunity, and they express the same membrane-bound antibody as the original naive B cell or parent cell. Plasma cells produce the antibodies with the identical antigen specifically as the antigen receptors of the activated B cells. However, they aren't membrane-bound. These secreted antibodies then circulate through the body. This selection of cloning specific B cells after identification of the pathogen is called clonal selection because the B cells are selected due to their specific variable regions on their membrane-bound antibodies, then cloned to be able to fight off the infection. Also, I'm drawing an elephant because elephants never forget, and this is a memory B cell. Before the body can generate an immune response, the antibodies and cells must move from the lymph nodes to the blood vessels to be able to circulate throughout the body. After antibodies reach the blood vessel, they can start binding to antigens and marking them in various ways for inactivation or destruction. For example, neutralization is when antibodies bind to viral surface proteins and prevent infection. Antibodies work with the proteins of the complement system to dispose of pathogens. These proteins increase the effectiveness of antibody-directed attacks on bacteria. A complement protein binds to an antigen-antibody complex on a foreign cell or enveloped virus, which triggers a cascade where each protein in the complement system activates the next protein, eventually generating a membrane attack complex. This forms a pore in the membrane of the foreign cell. Ions and water then flow into the cell, causing it to lyse and, therefore, die. The overall humoral response is very slow compared to the innate immune response. It can take days to establish. However, once humoral response has occurred, humoral immunity can last years and even decades.